In myeloma, we've learned a lot about genetics, mutations, translocations, and we are getting into like single-cell genomics and stuff like that. There's been less interest, or at least less number of studies dealing with epigenetics in myeloma. However, we know that indeed epigenetics is playing a role in the process of myeloma genesis, and part of the problem has been that the technologies to address some of the questions regarding epigenetics in myeloma have been lagging behind the techniques in genomics, and that has been one of the reasons why the amount of information is less. So my talk yesterday was trying to really get into high-throughput technologies to address questions regarding the chromatin structure in multiple myeloma. We've been working as part of the Blueprint Consortium in Europe, trying to address some of those questions. How is the chromatin structure in patients with myeloma? And what is the difference between this structure and the structure in normal plasma cells and also in normal B-cell differentiation? So from a very broad perspective, what we've tried to do is to interrogate all the different marks that define how the chromatin is structured in a cell, from like marks that give us information about whether the chromatin is open or closed, whether the genes that are in that part of the DNA are going to be transcribed or not, and what type of structures of the chromatin, what type of what we call chromatin states are in myeloma, unlike in other normal plasma cells. So that was kind of my general talk. I also gave a small introduction about what we know now about epigenetics in myeloma, because we do know fairly number of things. We do know how DNA methylation is in myeloma versus normal plasma cells. We do know also that in myeloma there are a number of genetic alterations, mutations that affect genes that are implicated in epigenetic regulation. But as I said, we know less about how the general chromatin is structured. What we've done is, as I said, interrogate different marks, different histone marks in patients with myeloma, and we have found that there is a very clear picture in myeloma where you can see that there is a huge activation of the chromatin. So instead of having like large pieces of heterochromatin in myeloma, we are seeing large areas of the chromatin that become activated in myeloma cells, and that actually has a lot to do with transcription. So probably those areas are being transcribed and are responsible for a number of transcriptional alterations that at the end of the day are going to be causing or participating in the development of myeloma genesis.